Hello dear students, I welcome you all on online lecture series, an initiative by Progressive Education Societies, Modern College of Pharmacy, Nigdi, Pune. I am Dr. Smita Pimpre, Professor at Modern College of Pharmacy, and today I am going to introduce you to the subject that is physical pharmaceutics. This subject is at B Pharm third semester, and today we are going to discuss about the content of syllabus according to Savitribai Phule Pune University 2018 pattern. Subject code BP302T Physical Pharmaceutics 1. Allocated hours 45 hours. Now, talking about the scope of this subject, this course deals with the various physical and physicochemical properties and principles involved in dosage forms and formulations. Theory and practical components of the subject help the student to get a better insight into various areas of formulation research and development and stability studies of pharmaceutical dosage form. The objectives: upon completion of this course, the student shall be able to investigate and apply various theories, laws, equations related to different states of matter, distinguish the principles of complexation, protein binding, and to use them for calculations of drug release and stability constants, and demonstrate use of physicochemical properties of drugs in the formulation, development, and evaluation of dosage forms. So overall, students, in the first year of B Pharmacy, you must all have introduced to different types of dosage form, their classification, the basic manufacturing procedures, the excipients, etc. Now in this year, Physical Pharmaceutics, this subject will teach you how the basic principles and concepts of physics, chemistry, and physical chemistry can be applied to the practice of pharmacy. Students, pharmacy is an applied science. Now, physical pharmaceutics is a subject which is going to attempt to predict the solubility, stability, compatibility, and biological action of drug products, and all these parameters will be studied with respect to physico-chemical properties of drugs and excipients. So, coming to the contents of the syllabus has 5 units. Unit 1, it is solubility of drugs. Under this topic we are going to study solubility expressions, mechanisms of solute-solvent interactions, ideal solubility parameter, solvation and association, quantitative approach to the factors influencing solubility of drugs, diffusion principles in biological systems, solubility of gas in liquids, solubility of liquids in liquids, solubility of solids in solids. Binary solutions, ideal solutions with respect to their colligative properties. Then Raoult's law, real solutions, partially miscible liquids, phase equilibria, phase rule, 1, 2 and 3 component systems, ternary phase diagram, critical solution temperature and applications, distribution law, its limitations and applications in pharmacy. So it is a huge unit which is majorly related with the solubility, and solubility is very much important property with respect to final therapeutic effect of the drug. So this unit has to be studied in 3 major parts, that is solubility, phase rule, which studies coexistence of phases, and distribution law or partitioning phenomena. Then unit 2, which is about states of matter and properties of matter. In this we are going to study different states of matter, how the transitions in state of matter takes place, latent heats, vapor pressure, sublimation, critical point, eutectic mixtures, gases, liquefaction of gases, applications of liquefaction in dosage form, that is aerosols, inhalers, relative humidity, liquid complexes, liquid crystals, glassy states, solid crystalline and amorphous states. Then methods of crystal analysis, X-ray diffraction, Bragg's equation, and polymorphism. Under this, the definition of polymorphism, different shapes of polymorphs, examples, pharmaceutical significance applications, and introduction to detection techniques of polymorphism. The part in this unit is also physicochemical properties of drug, where you are going to study refractive index, optical rotation, dielectric constant, dipole moment, dissociation constant, determinations and applications of these property we need to cover. Unit 3 is about surface and interfacial phenomena. The concepts explained in this unit are very important from formulation point of view when you are concerned with formulation of biphasic systems like suspensions and emulsions. So, subtopics we are going to cover under this are liquid interfaces, surface and interfacial tensions, surface free energy, measurement of surface and interfacial tensions, spreading coefficient, adsorption at liquid interfaces, surface active agents, HLB scale, that is hydrophilic lipophilic balance scale, solubilization, detergency, adsorption at solid interface. Next is unit 4, that is on complexation and protein binding. Here we are going to cover introduction, classification of complexation, applications, methods of complexation analysis, protein binding, complexation and drug action, crystalline structures of complexes, and thermodynamic treatment of stability constant. This is also an important chapter where we are finding out the application of complexation in pharmacy, that is either to improve the solubility or to improve on stability point from stability point of view, or even after administration of the drug during circulation the drug binds to various plasma proteins, and how does this complexation affects the therapeutic effect of the drug. Now next is unit 5, that is about pH, buffers and isotonic solutions. Here we are studying Sorance's pH scale, pH determination, electrometric and calorimetric methods, applications of buffers, buffer equation, buffer capacity, buffers in pharmaceutical and biological systems, and buffered isotonic solutions. This is all about what we are going to cover in theory. Now the reference books for the theory part: Physical Pharmacy by Alfred Martin. Now I have put a picture of the front page of this book, Martin's Physical Pharmacy and Pharmaceutical Sciences. Now this is a standard reference book which you can get, and I will recommend this book for all students to study from this book, because this is just like encyclopedia for physical pharmaceutics subject. Other books which are recommended are Experimental Pharmaceutics by Eugene, Tutorial Pharmacy by Cooper and Gunn, Pharmaceutical Calculations, then Lieberman-Lachman Pharmaceutical Dosage Forms for Tablets, Volume 1, then Pharmaceutical Dosage Forms for Dispersed Systems by again Lieberman-Lachman. Along with this, you can also refer for some chapters Physical Chemistry by Behel and Tully, and this book is specially helpful for studies of Unit 1 and Unit 2. Students, the purpose behind keeping this subject at B Pharmacy 3rd semester level, it is to provide a basic knowledge of physical pharmacy, pharmaceutics and biopharmaceutical principles as they apply to the development and assessment of various types of drug delivery systems, and to develop critical thinking and problem solving required to address problems related to dosage form design and its effective use, and to acquire technical vocabulary to discuss the pharmaceutical problems. Now I am going to brush upon some intended learning outcomes which are desired. Related to knowledge and understanding, by the end of this course, the student should acquire following, that is, physical principles of states of matter and phase rule, basic understanding of physical properties of solutions, dispersed systems, rheology, basic understanding related to physicochemical properties of drugs, including solubility, distribution, adsorption, and stability, knowledge about pharmaceutical colloids, understanding about pharmaceutical applications of various physical principles such as crystallization, condensed systems, and phase diagram. Intellectual skills the students will get are applying information regarding physical principles in designing dosage forms, recognize basic rules and equations regarding physical principles essential for pharmaceutical applications, and they will be able to apply rationally the principle in choosing adjuvants, excipients used for delivery and in formulation of biologically active molecules. Students will acquire certain professional and practical skills. That is, by end of this course, they will be able to operate different pharmaceutical laboratory instruments used in determining various physical properties, for example, surface tension, viscosity, adsorption and partition coefficients, etc. They will be able to perform skillfully some laboratory processes needed in pharmacy practice, such as determination of physical properties of solutions and suspensions, and they will be able to employ proper documentation systems. Some general and transferable skills I need to mention here. They will be able to work effectively in team to measure and understand various physical properties of solutions, calculate properly, express the solution concentrations, manage his and her time effectively, which is very essential, demonstrate critical thinking, problem solving, decision making abilities in variety of theoretical and practical situation in pharmacy practice. They will prepare themselves to discuss scientific reports related to studied topics. Now coming to the syllabus for practicals for the subject Physical Pharmaceutics 1, the code is BP306P. It's 4 hours a week. All the practical or the experiments are based on the content which is being covered in theory part. So the experiments are determination of solubility of drug at room temperature, determination of pKa value by half neutralization, Henderson-Hasselbalch equation, determination of partition coefficient of benzoic acid in benzene and water, determination of partition coefficient of iodine in carbon tetrachloride and water, determination of percent composition of sodium chloride in a solution using phenol water system by CST method, determination of surface tension of given liquids by drop count and drop weight method, determination of HLB number of a surfactant by saponification method. Eighth experiment, determination of Freundlich and Langmuir constants using activated charcoal method. Determination of critical micellar concentration, that is CMC, of surfactant. Determination of stability constant and donor acceptor ratio of PABA caffeine complex by solubility method. Determination of stability constant and donor acceptor ratio of cupric glycine complex by pH titration method. Determination of refractive index of given sample, and the last, determination of thermodynamic parameter using solubility studies. So overall 13 experiments are there in the syllabus. So this is all about the theory and practical syllabus of Physical Pharmaceutics 1. Now with this I end this video. I wish all the students best of luck and happy learning.